Thanks very much for the introduction and for the invitation to give a talk here. We heard a lot about cellular changes, and I will zoom out a bit and talk more about global connectivity between brain regions. If we look at connectivity, we can see something like this. So this is human brain connectivity. You can see fiber tracks between different brain regions. This is based on a non-invasive technology called diffusion tensor imaging. So you can put someone into an MRI scanner and you can then reconstruct fiber tracks in the brain. The color tells you the local direction. So you can see the green fibers going from the back to the front, the blue fibers going from the top to the bottom, and the red ones going from left to right, or in the opposite direction. So unfortunately, we don't know the directionality of those tracks. So you can see the spinal cord fibers there, the corpus callosum, and you can see the parallel fibers for the cerebellum. Based on this network, we can already say something about cognition in the subject. We can predict the IQ plus minus 3 IQ points and the age plus minus 8 years if we go from age 4 to 80. So we can look at the network and make predictions about individual subjects. And what we want to do in my lab is basically using connectivity as a starting point to make predictions how to treat patients like schizophrenia and depression patients. So can this kind of connectivity tell us something about how to treat patients? When we look at connectivity, we can distinguish different types of connectivity. We can look at structural connectivity, and this is real physical connectivity between different parts of the brain. It can be a fiber track between brain regions or an axon between individual neurons. We can look at functional connectivity, and this means that you have a similar activity pattern in different parts of the brain. So you can stick an electrode into two different parts, or you can look at the fMRI signal in different parts, and you then measure the correlation in the time series that you're getting. And if there's a very strong positive correlation, you would say that there's a functional connection between those two areas. And finally, you can look at effective connectivity. And this means that there was a causal relationship in the activity pattern. So first, region A becomes active, and then region B becomes active. So it can be that if A has increased activity, B has increased activity, or it can go in the opposite direction. If A is increasing, then B is decreasing. So if you have this causal link, you would say you have effective connectivity. So what happens during the lifespan? We can look at how the brain is changing during development and how the brain is also changing during evolution. If we look at the lifespan, this is just one example study by Chong and colleagues, where they looked at functional connectivity. So they looked at functional patterns in the brain, and they observed changes in young subjects and elderly subjects. So young is 24 and elderly is around 68, 66. And you see changes in global efficiency. So this means how easy information can travel through the network at the global level, and local efficiency. If you have one brain region that is connected to other brain regions, how well can the neighbors talk to each other, the ones that are directly connected? So you see changes for the elderly. You see a reduction in functional connectivity, and it's very tempting to link that to cognition. So maybe some cognitive deficits we see in elderly subjects could be linked to changes in brain connectivity. What you see in the lower half is the same network measures, but then plotted for individual subjects. So on the left, you have the first measurement, and on the right, you have the second measurement. And if you learn one thing from this talk, is that this is called a spaghetti plot. So this is one example of how the brain is changing over time. And the hope of understanding those changes is to get an idea what are the mechanisms leading to network changes, and what can we do to improve networks over time. So we can look at different features of the network. We can look at brain connectivity. We can look at cortical thickness, gyrification, and we can observe how that changes over time. And we can observe how it changes during healthy development, but also how it changes for new developmental disorders. And then the hope is to understand what mechanisms are involved, what connections are involved, and trying to improve the condition of individual subjects. And we're very interested in using brain stimulation in order to do that, rather than pharmaceutical drugs. So when we observe those changes over time, what are the features that we are looking at? So there was this discussion yesterday, how similar are different species. So coming back to that, we see a similar architecture in different species. If you just have a network, so let's say you have 100 parts of the brain, and then you basically represent connections in the network, it's difficult to say what species it is, because they all look very similar. We have similar patterns in terms of the topology of the network. So whether we look at the fruit fly, whether we look at the mouse, or whether we look at the rhesus monkey, they have similar architectures in terms of the network. One feature we find is that even though there was a preference for short distance connections, there were quite many long distance connections in the network, and there were more than would be expected. So I'll come back to that later on. Another important point is that we have imbalanced connectivity. I mentioned earlier that for fiber tracks, we don't really know how imbalanced they are, because there's limited data in terms of in invasive track tracing. But if we look at other species, the fruit fly, for example, we know that quite a lot of connections are unidirectional. So you have a lot of one-way streets in the network. So this is something that is very important for the dynamics in the system. If you don't have imbalanced connectivity, then you only have one parameter you can vary. Okay, so you can only change the overall strength. But basically, in this case, you can say that one connection is very weak or even absent, which makes a big difference in terms of the dynamics. Another feature that we see in all those networks is a modular organization. A modular means that you have many connections within a module and few connections to other parts of the network. So for example, many fiber tracts within the visual system, but few fiber tracts to other parts of the brain. We also have a scale-free and rich club and hub organization. And this means that some nodes in the network are more highly connected than others. So you might have, on average, 15 to 20 fiber tracts for each brain region, but then you have some brain regions with more than 50 fiber tracts, so they're more highly connected. And this is particularly true for subcortical structures, hippocampus and amygdala, structures that develop early on during development, but also early on in terms of evolution. We also have a rich club architecture. So if we look at those hubs, so those are those two red nodes over here. Oh, there's a laser pointer. Okay. If we look at those hubs, there is stronger connectivity between those hubs than would be expected. And this makes it easy for information to propagate through the network. So you can easily, let's say, integrate information within this module, and then information can travel to the other module here and be quickly distributed in the other module. So it's helpful for information processing. So what happens on the evolutionary scale? If we look at simple nervous systems, like in Cnidaria, you have different stages. You have the polyp stage and the medusa stage. For the polyp stage, the animal is basically attached to the seafloor here, and the whole function is to stream water into the interior of the animal here. So all you get is basically a contraction of muscle fiber and contraction of the animal. So what you have in terms of a network architecture is a nerve net. So you have nerve cells that are on the outside of the animal, and nearby nerve cells are connected. If you represent this on a circle, you have this kind of architecture here. So you can see that nearby neurons are connected. You don't have any long distance connectivity in a nerve net here. This is another representation of the same network. So this 15, let's say 18 over here. So this black dot means that there was a connection between neuron 15 and neuron 18. But then you can also have networks that have a functional organization where you have different functions in the system. And this is something you see in C. elegans, for example. You have ganglia in C. elegans, so you have clusters of neurons that are spatially separated. They form topological clusters, and they have different functions in the animal. So if we have a modular network, this is just schematic. So you have one module at the bottom with more connections within the module and one module at the top. If you generate lots of networks and then you average, you see that there's a higher density of connections within those two modules and then a lower density between those two modules. But we can even have further specialization, and this is a hierarchical organization. In the rhesus monkey, for example, we have the visual cortex, which would be one module of the brain. But within the visual cortex, we have two submodules. And those submodules correspond to the ventral and dorsal pathway. So you have one pathway for object recognition, another pathway for position and movement of objects. And this is represented in the network. So we get this hierarchical organization. You have two modules, but then within those modules, you have smaller submodules. If we average over lots of generated networks, you still have your two main modules, but you can see that you have two smaller modules within, and then again, two smaller modules here. So we have this hierarchical modular organization that we see in those networks. So how do we generate those patterns? How do we generate modules and hubs in the system? And rather than looking at genetic factors initially, I was thinking about what are like mechanical factors. So what are physical factors for establishing connections, and what are factors like timing to establish connections? So we can look at different possibilities, what roles are there in the system. So let's look at spatial and temporal features. And let's start with the formation of hubs. And we identified two mechanisms that can lead to highly connected nodes in the network. The first one we call old get switcher rather than rich get switcher. And the basic assumption is that the network is developing at different stages. So it's not like that all neurons and all areas are there at the same time. It's a sequence of events that is going on. So we have this time axis here. Some parts of the network are generated early on, and other parts are generated late during development. So what can happen is that those older regions, they can establish connections to those regions here. They can't establish connections to those regions because they're not generated yet. Okay, so they're not born yet. On the other hand, if we look at those younger regions, they can establish connections to all the existing parts of the network because all those nodes exist already. So what we would expect based on this sequential formation of nodes in the network is, on the one hand, that older nodes are more highly connected. Okay, so those older nodes, they can get connections from all the rest of the network, whereas the younger nodes can only get connections from the same developmental stage because other parts of the network have already finished establishing connections. And the second expectation is that older nodes receive more incoming connections. And we can basically test whether this is the case in the experimental literature. For C. elegans, we have some idea when neurons are born. So we know when are they able to form connections. And we can then look at the birth time of the neurons. So you have your 302 neurons here, and then the degree, the number of axonal connections, the number of other neurons that a neuron is connected to. And if we look at highly connected nodes, they're all born early on. They're all over here. So it seems to be the case that older nodes in the network have the ability to become more highly connected than younger nodes in the network. The second prediction was about older nodes getting more incoming connections. It is a bit hard to say. So this is based on the COCOMAC database. And the limitation is that not all directions were tested in the database. So the real picture might be slightly different from what you see here. But we see this tendency if you look at different parts of the brain. So those are evolutionary older parts, but also parts that are starting to be generated earlier during development. We have a slight imbalance, so more connections going to older parts. We have a slight difference here between frontal, temporal, sensory motor, and parietal occipital. But if you look at the newest parts of the network and the oldest parts of the network, you have 66 fiber tracks in this direction and 100 in the opposite direction. So it seems to be the case that older structures get more incoming connections, as we would predict based on sequential development of the network. So this is one mechanism that can generate highly connected nodes and a higher proportion of incoming connections. There was an additional mechanism that we discovered, and that's accelerated growth or nonlinear growth. So rather than adding one node to the network at a time, you basically have this pattern like a nuclear chain reaction that you have one node, it's dividing into two nodes, that node is dividing, and so on. So at each time step, the number of new nodes that are added to the network is increasing. So it's not like one node is added at a time. Yes. So it's basically the starting point and the duration of development that is different. Yeah. I think Georg would be the person to talk about evolution, but so I'm not sure about the. I mean, there's only a slight difference between those here. I think the really big difference is between neocortex and archicortex and paleocortex. Okay. So if we have this nonlinear growth, if we have an acceleration of the number of nodes that are added at each time, and we look at the distribution under those two constraints, if you have this regime here, so the wet one where you add one node at a time, you get a very narrow degree distribution. So you have an average degree for each node in the network, average number of connections, and then some have more and some have less. But if you have this accelerated increase in the number of nodes that are added at each time step, you have more nodes with few connections. But more interestingly, you have more nodes with many connections. You have a wider distribution. So you get more highly connected nodes if you have this nonlinear growth in your network. There's an additional mechanism that can result in highly connected nodes. So then we can think about long distance connections and bidirectional links. So what are mechanisms generating long distance connectivity? And the challenge for long distance connectivity is that it's very costly to establish if you think about a large brain. You have long distances, you need guidance cues, so you need a lot of molecular mechanisms, a lot of genetic circuits in order to be sure that you get to the correct target. Basically, the standard way of thinking about it is that this is what is happening. So in order to get long distance connectivity, you have guidance cues, and then axons are turning and moving around and finding the correct target. Before I get to that, let's look at axon growth in general. If we look at the distribution of connection lengths, we have this kind of relative frequency. So this would be in red area 17, just at the very local level. You have lots of axons with a low length, and then some axons are longer distance at this local level within one cortical area. And one explanation for this kind of exponential decay over here could be the explanation on the right-hand side. So the idea is that the neuron is growing in a straight line, and it's like you walking through a room. So let's say you start at one end of the room, and you're walking through this room here. And at each step, you have a certain probability to bump into someone else and establish a connection. So it, of course, depends on how many other neurons are there. But let's say in this example, this neuron establishes an axon, gets into this unit area. There's no other neurons, so no connection is established. It moves into this area, still no neuron there. And then it moves into this area here, and a connection can be established. So the probability to establish a connection after n time steps, where n can be time, but n can also be the spatial distance until you had hit another neuron, is 1 times the probability to step into a unit that is filled with a neuron, p, and then n minus 1 times the probability that you have an empty space before that. So that's q, 1 minus p. So this equation basically gives you this kind of exponential decay. So it can explain why it's more likely to establish a connection early on. So if you're thinking about moving into a room, let's say this room here, it's a lot more likely to establish a connection within 2-3 meters than going all the way to the other end of the room without bumping into anyone. Okay, so you have this exponential decay in terms of establishing connections. And growing in a straight line is preferential for axons. So we can have a computer simulation here where we have axon growth either in a straight line, or we allow the axonal growth cone to turn around to different degrees. And then we can measure how long does it take until this axon is finding another neuron. And the more you allow a turning of the axonal growth cone, the longer it takes until you find another neuron. And the reason is that the growth cone is basically moving into areas that have already been searched before. Okay, so the growth cone was going through that unit space. There was no neuron there, and the axon continues to grow. But because it can turn around, it goes through the same unit space again. And that's the reason why, on average, you get a longer length for those axons that are allowed to turn around. Yeah. Yes. Yes. A very, very simple model. Yes. Yeah. The other question is about how you establish long distance connections. So before we already saw it's a bit difficult to establish a long distance connection because you might just bump into things along the way. But it's also difficult because you might need a lot of guidance cues in order to get there. And we had a look at C. elegans. When can connections be generated in C. elegans? So you have the discrete time steps in C. elegans, and then the percentage of connection pairs present. And a connection pair can be present if both neurons are present. So if you know in the adult that 2 neurons are connected, and you see that both neurons are present at a certain time step, you say this can be the earliest time that those neurons can connect. So we can look at what proportion of connections is established at different time steps, and we can distinguish short connections, medium length connections, and long connections. And the gray bars here would be just randomly establishing connections. Okay, so if there's no pattern, then we would see this distribution here. But if we look at the actual pattern, both short distance connections and long distance connections are established early on during development. And the benefit is that you can save the guidance cues. So you might have 2 neurons that are spatially nearby early on during development. They establish a connection, and then in the adult, they are further apart. So it's one mechanism to save energy and genetic encoding by generating connections early on during development and then still having long distance connectivity. It also makes a difference whether parts of the network are established at the same time or sequentially. So we can look at a computer simulation where we have sequential development. So basically, you have one neuron, it's sending out an axon, then another neuron is sending out an axon, and so on. It's like one neuron after the other. So that's the extreme case. And then we have the other extreme case where all the neurons are there and they all start sending axons at the same time. It's basically just 2 extreme cases that you can look at. And we find that this makes a difference in terms of the network that you are getting. So if you have this sequential establishment of connections in your network, you have this distribution of connection probability and distance between neurons. So basically, this would be a long distance link. And you can see that you have more long distance links if you have a sequential process of establishing axons. We can look at the percentage of bidirectional links. So if we have this random growth in our model, how likely is it that two neurons have a bidirectional link between each other? And this probability is lower for serial growth and much higher for parallel growth. So we have some hypotheses that we can generate out of this model. First, that early starting neurons are more likely to become highly connected, because that's something we also see in those simulations, that they have higher local efficiency, that they have more long distance connections, and also that there were more bidirectional connections if you have parallel growth and fewer long distance connections. And we find exactly that in C. elegans connectivity. So in C. elegans, we can basically look at different time windows during development. So some neurons develop during the same time window and some develop in different time windows. And this is matching the expectation in terms of, are they more likely to be bidirectionally linked, and are they more likely to be highly connected? So this matches the model simulations here. We can also look at the formation of modules. So what mechanisms can lead to modular architecture? If we look at the modular architecture, it's a hierarchical modular architecture. So normally you would say you have brain connectivity, and then you have different modules like visual cortex, auditory cortex. But you also see a modular organization within cortical regions. And now I have to try to stop this at the right time. Okay. So we're basically zooming in into one hemisphere, and then we can zoom in into different cortical areas, and hopefully, oops. Okay. Just try again. Just have to keep the finger on the mouse here. All right. So what you see here is one example. So we look at connectivity within one cortical area, and we see a modular architecture. You can see you have two big modules within that particular region. And this is visible for many brain regions. And the next question is, this is work by Peter Taylor, who's now faculty in Newcastle. And the question is this just a random pattern based on diffusion tensor imaging? Because we know there are potentially lots of artifacts of diffusion tensor imaging. And in order to check this, we looked at connectivity between brain regions. So we have the left superior frontal cortex and left superior parietal cortex. We have connectivity within those two regions. You can see the modular architecture. So those boxes are different modules within those regions here. And we can then look at connectivity between regions. And if this is some kind of local artifact that happens close to the white matter, gray matter border, for example, we would expect that there is just some random pattern between regions, that there should be no organization of it. But what we find instead is that one module in one region, so that's this band here, is connected to one module in the other region. So you find a very specific organization, that a module in one region is connected to another module in another region. And those modules also have a spatial organization. So you can see that each module, this is for the left superior frontal cortex, basically corresponds to spatially adjacent patches of cortical surface area. And we're currently testing whether those modules directly correspond to functions as well. So if you think about the motor cortex, there were patches for arm movement, patches for leg movement, and we're currently looking at an fMRI data set to see whether those structural modules we see based on brain connectivity do correspond to functional modules in humans. So this is a hierarchical modular organization. And how can we generate those networks? And again, one way to generate them is to have different time windows during development. So in this model, we have two basic assumptions. One assumption is that you have different time windows during development. So you have three groups of neurons. some develop early on, some develop in the middle, and some develop late during brain development. That's one assumption. The second assumption is that the development is guided by having the same lineage during development. So basically, let's say you have a starting point here, cells are dividing, and they're all originating from the same cells, so they have the same kind of pattern when they develop and when they start establishing axons and so on. So with those two assumptions, having time windows and having spatial clusters, we do get network clusters as well. So you can see one module here, one module here, and one module here. You also see some overlap between the modules because you do have an overlap in the time windows. So based on having time windows, we can generate a modular architecture in the network. But modules are found everywhere. So in all biological networks and social networks, you have modules. So another question is, what is special about the modular organization of the human brain? And for this, we looked at networks that are artificially generated and have the same modularity as the human brain or the C. elegans brain, or they have higher modularity or lower modularity. This is work by Jin-Sop Kim, who's now a faculty in Korea. So we have the original network, and we can generate benchmark networks with the same degree of modularity, even though it looks very different. But if you calculate modularity, you get the same value. Or we can generate networks with a higher modularity or lower modularity, and we can do the same for C. elegans over here. And then the question is, what is actually changing in the network when we have a different modular organization? And one parameter we look at, I call it dispersion, is the degree to which one node is connected to other modules in the network. So that's the definition here, the number of modules to which node I links to, divided by the total number of modules. So for C. elegans, individual nodes would be neurons, and larger modules would be the ganglia in C. elegans. And for the human brain, there would be 1,000 regions of interest, and the higher organization is 66 brain regions. And just to show what this means, so let's say we have this particular node here, and this node is connected to other nodes in the same module and to nodes in another module. So it's basically connected to 2 modules out of 5 modules, so that would be a ratio of 0.4. So this node is connected to 40% of all existing modules in the network. On the other hand, the same node could be connected to all those other modules. So this node has the same degree, the same number of connections, but in this case, you get a very high dispersion. So the connectivity is distributed over the whole network. And we can then observe what happens when we change modularity. So this is the original network. This is the same network generated as a benchmark network, the same modularity score. And this vertical line corresponds having the same modularity score, and then you can go for a lower modularity and a higher modularity. The line here shows you values in the computer model if we generate benchmark networks with different amounts of modularity. And this is the actual modularity, dispersion in this case, that we find in the human network. And what this means is that given this kind of modularity, you would expect a relatively high dispersion. So we would expect that there were lots of links all over the place, but this is not what we see in the actual network. And one reason for this could be fasciculation. So it could be that you have early fibers, then other fibers are following those early fibers rather than randomly going in different directions. So it might be an explanation why we see this kind of pattern. We can also look at the characteristic path length, and this tells us how easy information can travel between different nodes in the network. And what we see here is, given the modularity we see in the human brain, the path length is actually higher than what would be expected for the modularity. So this means that the brain goes to some effort to increase the path length. which might be a bit counterintuitive. You might think that, well, the lower the path length, the better, the easier it is to go for the network, the better. But of course, it's not always good. So it's very easy to go for the network. It's like an epileptic seizure, activity can just spread quickly without any limits. And there is a benefit of reducing the spreading, because you can basically focus on auditory processing or visual processing. So it makes some sense to have a bit of separation in the network. And this is what we see here, that there is more separation than would be expected given this modularity. So what is the role of genetic factors? So this is based on the human picture I was showing earlier. It's the same network. And we look at something which is called algorithmic entropy. So how much information is needed to encode for the network. So let's say, hypothetically, you would have a brain where everything is connected to everything else. So if you have this matrix, it's just filled with ones. You always have a connection. And the algorithm is basically, well, you just put a one into every field. So you just go sequentially through each field in the matrix and you enter a one. And that's a very simple algorithm. But if you have a modular organization, it gets more complicated, because you put ones somewhere here, but then you have to stop. And you have a lower amount of ones between modules, and then you go back and you put more connections in again. So one way to quantify this is to compress the network. And this is basic compression you use on your computer as well. It's like a zip file. So you basically save your connectivity matrix in a CSV file, so you have zeros and ones. And then you basically compress that file. And you can look at the compression ratio. So how easy is it to compress the data? So the compressed adjacency matrix would be a bit like genetic information. So what happens if you compress the system? How small can the information get? Decompression would be like gene expression or Turing morphogenetic field. So basically taking the genetic information and generating your network again. And then the decompressed version of the network would be adult brain connectivity. And we can look at the human brain and C. elegans, and we can look at different modularities. And again, this line here is for the actual network. And if we look at the human brain in red here, so given this this pattern here for benchmark modularity, we have this compression ratio. But basically, we get a much better compression ratio for the actual network. Okay. So the original network in the human and in the C. elegans can be compressed a lot more than would be expected if we just have a network with the same modularity. So there might be some optimization to use fewer genetic rules in order to generate the network. And talking about generating the network, we had a discussion earlier. So what we find when we generate the network is that there was a lot of cell death. So almost half of the neurons are dying during early development, and they die during an early stage before birth. So normally you would say that there was reorganization in the brain, maybe because of input, because of learning and so on. But we see a lot of cell death at very, very early stages of brain development. So why would an organism spend so much energy to establish neurons and then kill them off later? And this is not only limited to complex organisms. We see that in C. elegans. C. elegans has more than 400 neurons initially, and then 109 neurons are dying, so you have 302 neurons left. So even in C. elegans, 25% of the neurons are dying during brain development. So why could this be the case? In order to study this, we developed a computer simulation of the formation of cortical layers. Within our computer simulation, we have different stages of development, and we have different probabilities. We have different probabilities for cell division. We have different probabilities to move to early apoptosis, and this early apoptosis is before the cell is migrating to form different cortical layers. So that's A1 over here. But then we also have late apoptosis, where a probability that the cell is starting to migrate and form different cortical layers. And once the cell is at the target layer, it's dying. Sounds very inefficient, right? But this is something we see experimentally, and we were able in this computer simulation where we have the proliferation zone where the cells are dividing and then they start to migrate and they form different cortical layers. We were basically able to reproduce the pattern we see in different species and in different parts of the cortex. So if you look at experimental values from the literature and our computer model, depending on the parameters, we were basically able to reproduce the patterns we see for different cortical layers in terms of numbers of neurons, cortical thickness, and so on. For human temporal cortex, red hind limb, mouse barrel cortex, and macaque visual cortex. And we were also able to reproduce different brain diseases. So you have polymicrogyria, you have other brain diseases, autism, and so on. And we can reproduce patterns we see for those diseases by changing parameters in our growth model. But what we also found in those models is that apoptosis is really crucial to generate those layers. So if you basically don't have apoptosis at all, if all cells are just continue dividing and migrating and form a cortical circuits, you basically are unable to generate the layer architecture we see experimentally. So apoptosis is crucial to get the correct layer architecture in different regions and in different species. And there might be a potential to use those kinds of models to understand what are the factors generating diseases, and those might be targets later on. So how are changed connectomes linked to cognitive performance? I'll just skip some slides here. So we looked at the rhesus monkey, and if you look at the distribution of connection lengths, you see this pattern here. For the human, you have fiber tracts that go to nearby regions, but also some long-distance fiber tracts. You see the same for functional connectivity, resting state functional connectivity, that you have some long-distance connections here. You see that for the macaque, a cortical network based on track tracing. And you also see that at the local level. So if you zoom into one cortical area and you look at the length of axons in this cortical area, you see a similar pattern with more short distance and fewer long distance connections. So the idea is that maybe reducing wiring is important. And this has led to the idea of component placement optimization, because it takes costs to establish connections and it's costly to propagate signals over a distance. So the idea proposed by Christopher Cherniak is that whenever you swap around brain regions, the total length should always be longer than before. So in this case here, you have this network, swap around nodes A and D, and you get a longer total wiring length as a result. And the idea was that this happens all the time, but that's not the case. So we have a reduction by 30% in the macaque, by 50% in C. elegans. So it's possible to get a better spatial arrangement. What is actually happening? If we look at the length distributions, that's length of a tract in millimeters, a number of occurrences. We look at the optimized network and we look at the original network, and you can see that what basically changes, there are fewer long distance connections. So when we optimize, we're basically getting rid of long distance connections, which on the other hand means that the real brain is like an optimal network with some long distance connections sprinkled on top of it, because long distance connections are really important. And they're important for several reasons. If you have long distance connections, you can have synchronous processing in nearby and far away areas because the signal arrives at about the same time. You have reduced transmission delays. So each individual synapse is introducing a delays in terms of propagating the signal, and you get around that by having a direct long distance link. You also have less cross-modal interference. So let's say you go from parts of the visual cortex, lateral and parietal area to frontal eye field. You can go there directly. You don't have to go to the motor cortex. And you also have higher reliability of transmission. So you can send a signal directly to the target, whereas if you send it over lots of intermediate steps, the signal might get lost. So whenever you have an action potential arriving at the synapse, you have a 50 to 90% probability that you don't have neurotransmitter release. There's a high likelihood that you will not have the same signal if you have lots of intermediate steps. And recently, we looked at this pattern in the human brain. So this is work by Chris Hayward and Shui Shen. And we looked at data from the Human Connectome project, 280 subjects. And it was possible to reduce the total wiring length by around 20%. Okay. So it's a bit less than the macaque monkey, but there might be reasons for that. In the macaque, we're looking at one hemisphere. For the human, we're looking at two hemispheres, both hemispheres. So that might explain this variability, but we're still able to improve the spatial positioning of regions. And of course, this is very different from what you would expect without component placement. You would expect that the distribution is over here on the right-hand side of the zero. So before, we thought that the reason for this organization would be those long-distance connections. They're really, really crucial. But for this paper, we also looked at dynamics in the network. So the original network is reducing synchrony and increases metastability. So let's go through that. If we basically have our brain regions, we can have simple oscillators, Kuramoto oscillators that have a certain frequency, let's say 40 hertz or 60 hertz or 80 hertz. The coupling between those oscillators depends on the strength of the fiber tract. Okay, so the stronger the fiber tract, the stronger the coupling. And we can then observe the synchronization in the whole network. And we can also observe the fluctuation of the synchronization. So this would be the mean synchrony. So this line here, just the average. And then you have the standard deviation, which is metastability. And if we look at those two cases, the original network and the one that has been optimized for saving wiring length, we see this pattern here. We find a higher synchrony for the minimized network. Okay, so there's more synchronization. And we find a reduced metastability in the minimized network. And this might have cognitive effects. If you have a higher metastability, the system is fluctuating more, and it's easier to get into different brain states. And what we found for dementia patients is, if we look at EEG, they stay in brain states for longer. So it's more difficult for them to process information, and it's difficult to just switch to a different state. Yeah. But basically, if you have higher metastability, it's easier to move to different states. So it might be beneficial for cognitive processing. So it would be another reason to have this kind of organization that we see in the human brain to enhance the processing in the system. So this already indicates that spatial features are very important for the processing. We also see that if we look at IQ. When we try to predict IQ in the same Human Connectome project subjects, you have different subscores of those cognitive tests. You have different network features like local efficiency, the clustering coefficient, using an unweighted and weighted network, edge density, a path length, an unweighted and weighted network. And then we looked at how those network features correlate with IQ. And we see some features that have a bit of a correlation here. But if we look at spatial features, we see a very strong correlation. So spatial features could be the convex hull, which is like the, if you wrap around a foil around the brain, you know, what would be the surface area. And also the distances in the system. So if this is region A connected to B and C, we can measure the distance between B and C with different configuration. And this all gives us very strong predictions of subscores of IQ. So this means that the spatial organization and the length of connections is really important. So I call that the delayome in the manuscript. So it will be important in the future to really look at delays in the system. And in the future, we want to use non-invasive brain stimulation. I'm establishing a lab for focused ultrasound at Nottingham University. There are some other systems in the UK. And the benefit is that you can directly reach deep brain structures. So in this example, you can directly increase or decrease activity in deep brain structures in the center of the brain without stimulating superficial structures. It can be very focal, so it could be up to a few cubic millimeters. but normally it's a bit more than that. For our systems, you can reach deep brain structure. It's non-invasive. It can be used for humans and rodents, and it's possible to measure MRI and EEG while stimulating, so you don't have any interference there. It's a novel area. 80% of the papers appeared in the last 2 to 3 years, and those are just examples of studies in humans. Just want to point out some studies, Sanguinetti, where you stimulate the frontal lobe and you can improve the mood of a subject. So both in healthy subjects and also in depression patients. And there are other studies here. There was one study in Shanghai on schizophrenia. This starting point, and we want to change brain connectivity. So we want to use network analysis and computer models to find out what parts of the brain to stimulate and then use focused ultrasound in order to change connectivity over the long term. So in summary, connectomes show modular organization across scales and across species. There are spatial, temporal, and genetic factors that can influence the development of network topologies. Spatial features, delays might be important or more important than topological features for brain function and cognition. And future research might be able to change connectomes to improve function for brain network disorders. And small advertisement, there's a book with more information about that, looking at models of brain development, looking at actual development in healthy development and different brain diseases, looking at the effects of learning and of brain stimulation on the network. I would like to thank my team in Nottingham, alumni of my lab, collaborators, and the funders. If you're interested in the topic, you can follow me on Twitter, and thanks for your attention.